after that post-translational modification. So we can identify proteins. We talked about using these different methods, and then we talked about, well, then we have to identify them and include, post-translational modifications into our consideration. So we talked about how to do that. And then we talked about, let's see what else, post-translational modifications, and then the last one was applications, right? Which was just an overview of what you can use this stuff for. I don't expect to have very much on that. That's not it at all. Okay. So, does anybody have any questions?
Is the length of the exam probably going to be the same as last time?
Yeah, probably about the same as last time. How long was it last time? Like 4 or 5 problems? I don't know.
It was 5.
It was
I don't remember.
5 and a
But I
bonus.
remember taking, but I hadn't have enough time to write everything.
Yeah.
Yeah,
I
it was,
wasn't here last time.
it was 5 problems with a bonus.
5 problems with the bonus.
Mm-hmm.
So about like 1 question per chapter for this one.
Yeah, that'd probably be about right, because we only have an hour. Let's see. There's a class before us here, so I can't really,
Yeah.
No,
can't
wait.
start early, but we could maybe go a
There's-
little
wait,
longer.
what?
What's that?
On Friday, this matrix class doesn't meet.
Oh, they don't meet?
Yeah, it's a Monday, Wednesday class.
Oh, okay. How do you know that?
Because I've been
Creep.
here before.
Oh, okay, okay. Well, why don't we just start at the normal time, but because I'll be here, I can allow you to go a little longer, because there's nobody after us, for sure. So last time, Miss Johnson, had other stuff to do, so they couldn't wait. All right. So I just wanna we're waiting for you, Chad. We're like, we cannot start without him. Because then we would just go into utter chaos,
Yes.
high entropy state that we wouldn't know what to do. We just-
Man, I got a pecan tomorrow. The entropy needs to die. Calm that down.
All right,
I need
so
to
I'm
inject
gonna
some
pull up
solution
a
into my
blank
veins.
slide, but I want to have my slides
You have a solution to make that.
ready to go here, regulation,
Did you finish that
for
one?
reference.
Nope.
All right.
How about you get?
I didn't finish chapter 5. I didn't start chapter 5 yet.
Translation modification.
I'm going to look over some stuff today, Joe.
What did you do all day yesterday?
Protein interactions,
Cool.
that was the
Busy.
other one. Looking for networks,
busy.
interactomes,
You were here till 3.
and
I was up to like 4. Oh.
You went to your nap too?
application. Okay, all right. So let me
Went to
pull up my
Whataburger like 2.
PowerPoint slide.
That's swamp by Jim Kim kids there. They got trackers on me. That's
How
not
far did you get that one?
How far did I get? I finished 3 and 4.
You did?
Yeah.
How long did 3 take?
Okay.
Not too long, about an hour.
Oh, really?
Yeah.
All right.
How long without the solution remaining?
6-7 hours. Jag helped a lot.
All right. So
Crying
we'll
helped.
start with translation chapter. What's important to know about translation? Well, we need to know things like the codons. We don't need to know the codons, but we need to know certain things about it that, there are codons. Where is my stuff? I won't draw it every, so we'll know about codons, right? 3-letter code that is required. A tRNA has the anticodon for a codon, right? There is a codon bias. Do you remember that? So we remember that there's a codon bias, that not all codons are equally found in the cells. And how did I say we could, the cell actually uses that. At first it was like, oh, it's a codon bias, you
Affects
know.
the rate.
What's that?
Affects the rate.
Affects the rate, right? So we talked about how we believe now. So we talked about codon bias. Affects rates.
Dr.
And
Patterson?
what else could it do? Sorry. Okay. So we talked about codon bias, we talked about rates, and what does that help to control in higher
The
organizations?
amount of protein
Yeah,
that's generated.
yeah. So we can control stoichiometry of proteins, right? So it helps with efficiency. So we talked about having, what did you have? You had alpha 2, beta 6, gamma 4. Well, what they're starting to understand is, so that's how many make up the complex, that the codon bias can actually bias it so it controls the expression levels of each of those subunits. That's right. There may also be other things like controlling the folding. If you get stalls, you know, because you have a low tRNA number, right, that will stall things, and that can also influence the kinetics of protein folding. Okay, that leads me into an important thing. Eukaryotes versus prokaryotes. What's the difference?
Where the ribosomes are located.
Yeah, so in prokaryotes, transcription is coupled to translation. So remember how this is, you have your DNA here, and then we have our RNA polymerase. Sorry, it's going to talk to me.
Is it all in one complex? Does it bind? Does it bind to the DNA
Yeah,
itself?
they are coordinated. So, sorry. So as, you know, this is being produced off, you know, kind of hooks around, and then we have our ribosome, right, which has two parts. Here's our ribosome, and basically from this we're getting our amino acids coming out. So transcription is coupled in that case. It's all done in the cytosol, and this is very different from eukaryotes. Right? Transcription is done in the nucleus, then the transcript gets exported, and translation occurs in the cytosol. So we have a difference here that in eukaryotes, right, we have the nucleus. And so transcription is occurring here. And then that has to be taken out. And that's our mRNA transcript. And then out here we have our ribosome. Okay.
Is there codon bias in both? In both
Well,
prokaryotes
it's
and
the
eukaryotes?
codon bias, yes, I believe so. There's a codon bias in both, yes. And because of that, you'll see, like, if you take coding from a eukaryotic gene and try to take that gene and express it in, like, a bacteria, it won't express, it might not express very well in the bacteria because of the codon bias, not just because of things like post-translational modification. Okay. And so we have those differences. There are other differences, but we use as our model, remember, the prokaryotes in understanding translation. So let me just put use I remember. So you'll want to know the different steps of translation. So there's an initiation, there's an elongation, and then there's a termination, right? Okay, each of these is controlled by a set of factors. So,
Those are transcription factors
Mm-hmm.
or translation?
those will be translation factors.
Oh, okay.
They're not actually, they're guiding the translation. The transcription factors led to the transcript being formed. But, yeah, they're regulatory proteins in a similar respect. So let's just get some general features. So you have to have initiation, you have to have elongation, and you have to have termination. Okay? The initiation is aided by initiation factors, IFs, elongation by elongation factors, and termination is aided by what we call release factors, RFs. Okay. And all the ones that we're talking about, because it's simpler, is going to be prokaryotes. Eukaryotes basically have a similar strategy, just different factors that are involved, more of them and all that.
So we don't need to look into.
No, let's just understand the basics. If you understand the basics in prokaryotes, you can understand eukaryotes. Okay, so there are 3 initiation factors, IF, 1, 2, and 3. Okay. There are different EF factors. There's EFTU. Sorry. There is also EFG is the main one. There's one more. EF, it's not TU, it's let's see what, I wrote it down. TU. There's one more that's involved with TU. That, and I forget what it's, I think it's TS, if I remember right, or something like that. And this one interacts with this one. But these 2 here are involved in the actual cycle. RFs, you have RF1, RF2, and RF3. Now RF3 is a universal one. This is what causes release. You either use RF1 or 2, and we'll talk about that if you want me to draw through it. Okay? But these are the main ones you have to know. Initiation factors, elongation factors, and then the release factors. So you guys want me to go through each one, and we can just kind of break it down and see how they work. Okay, initiation.
Could you go back real quick so I could? Okay, thank you.
Okay. So remember, we start out with a ribosome. Okay. Depending on which one, you have either 70S or an 80S. And that's made up of a 50 or 40S and the, I don't know, 50S and
It was 50 and 30.
30S, right? Because it's not. So you start out with those, right? Now, how does it open up to actually get the transcript? You have those 2 transcription factors, TF1 and TF3, that bind to and destabilize, okay, the ribosome assembly so that the 2 separate. So they bind. Okay. Bind to. So I'm just going to put 1, 3. Then if you have an mRNA transcript, right?
Which one's, so this, 50S and a 30S split?
Yep. And then I believe the 30S binds to the mRNA transcript. Okay? Aided by, we have the transcription factor, TF2, which is a GTPase. So it has GTP value. The GTPase, and once it binds, yeah.
Is the T a transcription factor or translation factor?
Oh, translation factor. I'm sorry, and these should be IFs. IFs, sorry. Initiation factor.
Oh, okay.
Sorry. Initiation factors. And then what happens now is that initiation factor binds, and then now when this binds, they'll come together. And what is the site that dictates where translation begins, or where it can start to look for the start
cap.
codon?
The activation.
Well, cap in eukaryotes, but we're talking about,
Is this like the A site?
yeah, so promoter, but where does this bind, where does the actual ribosome bind to? Do you remember?
That guy's.
This guy's name, right?
It's the
Shine Dalgarno. Yeah, so it binds
Think
to
of the
the
beard.
Shine Dalgarno, right? And then after the Shine Dalgarno, right, it will start looking for the start codon. So we bring that together. Now we have all these factors together, plus this, okay? And now this GTPase, once that brings everything together, it will hydrolyze. So, and we get initiation and our start. So
So clamps back down?
What's that?
It's the 50S and the 30S
Yep,
clamping back
clamp
down on the
clamp
mRNA?
back down.
Is
And
that
then,
TF2, is that initiation factors just come off?
yep, once it brings it together, it's bound to the Shine Dalgarno, it brings it together, and then that GTPase, which helped to bring in the other part, right, will hydrolyze, make GDP, PI, and then those other initiation factors 1 and 3 will also fall off. And basically now we have initiation. We have initiation by bringing in our tRNA. Okay, sorry. tRNA is brought in with IF2, brings in the tRNA. So that's how you initiate it, right? Initiation is started by the tRNA. Okay, that'll bring it together. And then what happens is the IF is bringing in the tRNA.
So then step 2 it brings it in?
Yes. So it's dissociated. It'll bind to it. This transcription factor will bring it in. Okay, it will bind, find the Shine Dalgarno, start, and then it will put in the tRNA, and then it hydrolyzes away. The tRNA is left behind. And it has a methionine, right, in that position, and it will be in that second site.
So it clamps around the tRNA in step 3? That's what you're showing?
I'm saying it's in the acceptor site. Remember, there are 3
So
sites.
after clamp, so it's
Yeah,
all
clamp
clamped
down,
So
and
it's-
then the GTPase, once it found the site and clamped down together, it hydrolyzes it, it leaves, and there it goes. I'm probably doing a truncated version of this from what your book says, but basically that's it, and that's the initiation process. And then this, of course, can be recycled, right, GDP. So let me see here. Let's make sure. talked about codon biases. We talked about initiation. So basically what I told you, that bind separates the two, okay? This will bind in here with that, find that Scheindahl-Garno, and then it basically brings the whole complex together, and when those touch, it hydrolyzes and leaves it in that A site, or the acceptor site. Peptidyl site. Acceptor, peptidyl, and the exit.
So it does this for every tRNA that's brought in, right? Does each one-
No, now then once it gets that charged, it's stuck on there, and then it's going to do the elongation.
Okay.
Does that make sense?
That's
So
what
that-
these are the
that finds-
initiation factors.
So that's the codon, then the anticodons come over for each one? that
Yes.
changes?
Yep. So this is our initiation. Those initiation factors, and of course this will have to be, recharged so that it can complete this. Okay. So Scheindahl-Garno finds that site bound, then it brings together those pieces, and what it's showing basically is once those connect, you do the hydrolysis and they all leave. Okay, so,
Dr. Patterson,
yeah.
would you expect us to explain the process, or do you want us to explain it as based on the pictures?
Explain it,
Explain.
just explain the process. You could draw a schematic to show it. That's fine. Okay. TS, I was right. TS. Just go through each of those. And that's how you do the initiation, basically. Do you want me to go through each step, or you just go through each step and you can draw out, practice drawing out, or just explaining in words what happens? Either way. Drawing it out would be better. Okay.
Words.
Words, it doesn't matter as long as you. Now, knowing the pathway is one thing and is important, but then knowing it's supposed to help you maybe make bigger connections about the pathway. So if you look at all the other processes, so let's talk about elongation. And the A is the acyl transfer site. So P is the peptidyl, right, site. A is the acyl transfer, and then E is the exit. So let's go from here. If we're talking about elongation, okay, that says elongation. All right, so what do we start with? We have it charged now. Okay, we have the P site, the A site, and the E site. And what starts out in the P site is our tRNA with methionine, right? Yeah, this is not.
So what were the sites again?
So-
E's exit, so.
That's
Peptidyl.
Peptidyl and acyl transfer. Because this one is where the peptide bond is actually formed. This is where we have the acyl transfer between the different tRNA. Okay, so let me just draw this a little bit different to make my tRNA, I'm just going to call it T. There's my T. Okay, it's already in this site. Okay, and then we have, of course, this, and that's running through there. Okay, so we have that there. Now, how does it get elongated? We have an elongation factor. Okay. TU. And this is a GTPase. So bound to it. And it will have bound to it also the next tRNA. Okay. And right, in order for it to come in here, right, we have to be able to have anticodon base pair. So we actually use that. Okay, it's going to place that in there, and we're going to get that site and that site. We have our T. Now that has methionine. And then here we have our next T. Okay. And I'll use a different color. That has the next one there. So this charge with GFP comes in, it aids in transferring that in here if it has the right codon, right? And once it puts it in there, it will hydrolyze this. And now our transcription factor has GDP bound to it, okay? But it's the same one, EFTU. And then there's a process of recharging this. I'm not going to go into that, but that other protein, basically there's an exchange protein that will come in, bind to this, exchange it with GTP, and allow it to get reprimed with another tRNA. So now we have this in here, okay, and then we get our connection. So now we form that peptide bond, but we have a problem here. We have to move everything, right? So that it will elongate. Okay. How do we do this? This is aided by some more elongation factor. Okay. And this is done by this another GTPase called EFG. Okay. And now the purpose of this is to aid in transferring it, right? Because we have to go from the A site to the P site and so on. So what's going to happen here? GDP plus PI. Going to aid in taking these, and I think I'm cutting out a step, but we have to get these things opened back up so that, right, this will basically help it to move from one site to another. So we have our T, M, here we have our T, connected. So it moved it over. And then what happens now? Well, that tRNA is no longer attached. So we go here and we've extended it. I think I'm keeping out, but that's the general process. You open it back up and then it goes.
So where'd the other tRNA come from? Because in the other step you only had two. Now you have two. You have one charge, one peptide, and a tRNA popped off?
Yeah. Because now they form the peptide bond, that tRNA will leave.
So it's the first tRNA that's gone?
Yes.
Okay, cool.
Yes. So. Elongation, yeah. Pretty much. So we go there, we get it in that site, okay. We have peptide bond formation. Okay, now it's connected, and then that protein pair EFG, right, using GTP. It basically helps to transition and slide that over. Interacts with the protein so that it can do a ratchet effect, and it allows that all to move over. Then that tRNA leaves. So they're showing the bond formation. Basically, it comes in, it leaves, and then the bond forms. I guess I kind of skipped that. You can put an extra step for that. So this extra step. That's fine. You know that it goes in, it gets attached, and then it does the bond formation. Same deal. Okay. Now, do you see something about the two processes, elongation and initiation, that have some similar component? GTPases. Do you remember me talking about why are GTPases important? What is
The
their
coupling of the reaction.
Molecular.
They're molecular clocks. So you see this over and over and over again, that what controls the whole process are these molecular clocks. And this is the same in signaling. And we'll see that the termination is the same way. So let's go back here and let's do a termination real quick. And I don't care if oops, sorry. I swapped it, you can see what. And I don't care if you draw in that extra step or you draw it in as one. It puts it in and bond formation occurs. They're going to occur pretty much in sync with one another. There'll be maybe a delay to that. So whether you do it 3 steps, 4 steps, I don't really care. Okay, termination factors. Remember there's 1, 2, and 3. 1 and 2 serve for different stops. Yeah.
So where was EFTS on elongation?
EFTS was the one up there that's recycling the EF2.
Okay.
So it's just up there, pulling that apart, letting it get rebounded GTP, and then letting it get recharged.
Does anything happen with the A site? We just do this, do we just run it back around at the next?
Yep, then it would run back around. A site's open for business, it goes in. So in the process, elongation factor TU, what's its purpose? To transfer the tRNA into the active site. It basically helps to change the conformation to allow it to come in so that when it base pairs, it will charge it. If it doesn't have the right guiding tRNA, it's not going to do anything. It's not going to make it in there. But once it gets the right base pairing, it basically hydrolyzes, releases, and leaves it in that site, and then that reaction occurs, the acyl transfer where we form the peptide bond. Okay? Then the second GTPase is downstream. Its purpose is to help move the tRNA over one. So what was in the P site goes to the E site. What was in the A site gets moved over to the P site. And then that tRNA can leave.
It is both?
Yeah,
If
yeah,
there's
it's
something in there,
ratcheting,
it moves
it's
everything
ratcheting
over
it so
one.
they move in kind of unison. They move over. All right, last one, termination. Like the Terminator. I'll do that. Okay. Termination. So TF1 and TF2 are for different stop codons. So there's TAA is a stop codon, and TGA, for instance, is a stop codon. And then there's another variant of this. These are the main ones, though. But there's TG. What are the other? I forget. You don't have to memorize them. TAA and TGA are the main ones. So these serve for termination of different stop codons. So what happens? Okay. These are our termination factors. So we're going along here. Here's our ribosome. Okay, and we got our tRNA in here. And right off of that, we've got this peptide chain that has been growing. Okay, but now we, for instance, reach TAA in the sequence. So these transcription factors will initiate termination. They'll identify that, come in and bind. To that. We'll just say TF1. So we have
Or two.
this. What's that?
Yeah, which one
Or
was?
two, either one. Will come in and bind here. So it's 1. Oh, sorry. Why do I keep saying TF?
Oh yeah, this one's R.
Release factor. I'm sorry. Release factor.
What's the thing on the bottom, in purple?
That's the stop codon.
Oh, so that's actual codons?
Yep, that's an actual stop codon. So it would bind to that. And then basically you have RF3 that comes in. It's a GTPase as well, so it comes in as GTP bound. And then when it binds to it, it will help release everything basically. So binding, we'll just show it bind and then release. Release factor 1, 3. Here's our GTP off of here.
So the RF1 binds to the stop codon?
Mm-hmm. And then it hydrolyzes basically that once it hits, and it causes destabilization of the complex.
So with the RF3 binds to the RF1?
Oh, and what it does is when it binds to this, it brings water into the active site. Sorry about that. So that the water now with the RF1 or 2 with RF3 brings water in there, hydrolyzes that, and releases it. So we get water that comes in, H2O. This will release our polypeptide, and then the release factors, RF1 will be released, and RF3 will now have GDP and PI. And now that's basically empty. It's destabilized, and now it can repeat the process. So again, GTPase is our clock for this.
So the whole complex destabilizes, the 50S and 30S will pop open?
Yeah, once you have that leave, it becomes destabilized. And then those initiation factors from the beginning, because it's destabilized, they'll come back in and moderate it so that it will dissociate, and then it can be ready for another one. But pretty much, you know what I say here, yeah. Binds there to that site, okay, one or the other, okay, and then we get our release because of, you know, the GDP is our GTPase is the TF3. They dissociate, okay, that allows it to be cleaved, everything goes off, and now it's destabilized because it doesn't have any tRNA in there, and it's at the end. So I think what we drew is pretty consistent with this. Okay. So those are the three processes. Going to be fairly similar, maybe a little more. There's going to be more factors associated with eukaryotes. a little bit more complex. But if you remember those, it all has the basic same kind of pattern. We've got initiation, elongation, and termination. There's just going to be more factors involved in the protein, in the eukaryotes. Let me just scroll through here. Eukaryotic differences. We've already talked about these. There are different places, right? So the other thing is trafficking. You know, that different proteins are trafficked in eukaryotes different. But what we were talking about doesn't matter. Cytosol is there. And usually things get trafficked based off of leader sequences, right, in the proteins. We talked about that.
Are they attached to cytoskeleton?
What's that, leader proteins?
Yeah, so the
Sequence?
proteins can be moved between,
Um,
or is it?
So they get put in clathrates, remember? Or not clathrates, clathrins. So you remember me talking about how you can transfer these. So you make the proteins in the endoplasmic reticulum or at the surface if they're going somewhere else other than the cytosol. And then basically things from the ER get transferred to the Golgi, and then from the Golgi, you know, they bud off and they can go to different places.
But everything else is concentration based? Because it's actually in the cell itself, in the cytosol?
Yes, anything that's targeted for the cytosol will stay in the cytosol. If there's a targeting for somewhere else, it will basically be packaged up into a vesicle, and then the vesicle will transmit it somewhere else, or it has a leader sequence that will transmit it into, say, the mitochondria or something. There's got to be a leader sequence that directs where it goes.
Because some of the cells are like, your nervous cells are almost a foot long, like, move the protein all the way down there. It's
A foot long, your cells?
Yeah, isn't it a single cell that makes up your nerves? Some of them? They just have multiple mitochondria across.
I'm not a neurologist, so I'll leave it up to you. But we've talked about kind of that. If it's going somewhere else, it can either be endoplasmic reticulum, or if it has a leader sequence, it can be trafficked into those other sites. It has to have something that tells it where to go, though. How am I doing on time? So, we made it through that. Could you do this? Could you answer things about elongation? All of that good kind of stuff. I'll send you out a review sheet. But basically, if you go through these slides, I know there's a lot. We talked about post-translational modification. We talked about 3, phosphorylation. We talked about ubiquitination. And we talked about, as it relates to proteomics, we also talked about, what was the other one? Glycosylation. So we talked about those. We talked about it here a little bit. Let's see. I will send out something on that. For each of these, I'll give you kind of a rundown. So proteomics.
So our test is basically going to cover chapter 24 stuff and on.
25, I think is what it is. 25, 26. Regulation of gene expression. So it's 25 is decoding. We've already done transcription in the last one.
Okay.
So one on decoding, translation.
Oh, yeah.
Probably one on regulation. Probably one on how do we investigate proteomics, thinking of mass spec. So let's see here, applications. So did I knock one out of here or something? I thought I had it. So we talked about the different kinds. So things that you'll want to know is MSMS. How does that work? So mass spectrometry. How do we use it? Predominantly we use ESI, electrospray ionization, and then MSMS is where you use it. Remember, you look at the parent molecule and then you send it through and knock the heck out of it, break it apart into fragments. And the idea is if you fragment a parent peptide molecule, you can either sequence it completely, or that's the idea, you could sequence it, or from the masses, you can use it to go back to the genome, right? Use bioinformatics and identify the protein. So understand that process of how do I use mass spectrometry to identify a protein. And we talked about that from beginning to end. We talked about how you can, separate them out, right? We want to separate them out, get the sample a little simpler to evaluate. Then we digest it usually, take the protein, cut it up into small fragments, send it through the mass spec. We get the parent molecule for that. And then we can send that parent molecule through again, do MS on it, right? Break it apart again, figure out its sequence, and then use that to go back to the genome, and then we can identify the protein, right, from that. So we use bioinformatics. We talked about that. So let me just, on this thing that we were doing, let's just summarize some of the things. So obviously I can't have you do all of the termination and all this because it took us an hour to get through it. So I'm not going to do that, right? So what should you know? What are some, sorry, what are some main things?
30-day points each.
MS, how is it used? Things like MS, be able to describe that, and how it's used with bioinformatics. There are some processes that we talked about, like when we're talking about looking at phosphorylations. What do we try to do? We try to separate and increase the amount of phosphorylated proteins. So we talked about things for separation and enrichment, right? That's kind of a common theme of certain samples. We focused on that quite a bit. And then using mass spec, we've talked about that. What else? I'll send you out a sheet here. Basically, yeah, it's going to be one question from each chapter, is what I'm thinking. I'll try to keep it more in line with the proteomics part of the course. What else did we talk about? Protein arrays. What is it? Could you describe a protein array? How does it work?
Is the bonus going to be something like, here's a mass spec, actually determine the
Hey.
amino acids?
Sure, you want that?
I'm good.
Protein arrays, what are they? So, one of the things that we talked about, remember, how do we different, the purpose of proteomics is really what's present, and then we can use that to figure out if we change the conditions, how has something changed? And basically, if you boil all the methods down, it's always looking at the difference between a disease state and a regular state, or a drug cell state versus the normal state, or something like that. And what's the main way that we determine that? Mass spec, right? We label the molecule in some way. We label one state versus another state. You remember that? So you either grow up your normal cells on regular media, and then the one you want to probe, you grow on heavy media. Remember that? Or you label them with different labels, one that's heavier than another, right? So we've got those two approaches of how do we use MS, right? So we have, say, a baseline state. We can think of that as normal. And we want to see how does that compare versus some change state. I mean, this is effectively what we've been talking about. So think of change state as disease, drugged, or whatever change you want to evaluate. And so the idea is you're going to have to differentiate the same proteins, okay? usually by mass. So you either tag with a different mass. So one would be light, this would be heavy in some way. And then you use mass spec, right? M over Z. And the idea is intensity. You look at the size of the peaks, right? It might be something like this. And this would be our heavy. And this would be, say, our light. And how do we figure out changes? By the difference, right? Difference in intensity tells you the change, right? We went through all that, but that's basically how you use mass spec and why it's so powerful. You can selectively label the same proteins, but from different states. You can label one with a heavy and one with a light, either by growing it in heavy media or by tagging it with some reactive compound. One is heavier than the other. And then when you run them out on mass spec, you can say, oh, this is what's happened to protein A. Protein A under normal conditions is elevated. Under these changed conditions, we see a drop in the intensity. So it's a way for us to look at relative amounts. Does that make sense? So if you want to know why does this person have this disease, you might find a protein that's linked to that disease. And then you can compare the two and say, oh, it's showing that there's a reduction in this protein. So maybe that protein is involved when it's reduced. It's causing that effect. And then you can go back and figure out, what is that protein to? What is its involvement in the cell? And then you can think about, well, how do I improve that? How can I drug a person to increase that? How can I change the person to get them away from that state that's disease? And that's the application, right? Does that make sense? Yeah, this is probably the worst prepared I've given you guys a review for. I'll give you a review, and I'm sorry I didn't get it to you sooner. But basically, looking at those kinds of, what are the common themes in each section? And can you answer questions about that?
Dr. Patterson?
I'll send you a review, yeah.
You said MS. Okay, the first MS is mass spec. Is the second MS?
Yeah, so MS means you've sent it through, you look at the mass of the parent molecule.
Oh.
Then what you do is you use the mass spec to break that parent molecule apart. And then you see what's the mass spec of the mass spec
Gotcha.
peptide. And then you can break it apart into all these fragments.
Okay.
So I'll send you this. Save. So let's just make it kind of simpler for you. I'll ask a question on mass spec because that's so central. Okay, mass spec related to proteomics. Okay, I'll ask you a question related to the codon bias and translation. It might involve the codon bias, or it's one of these parts of translation, just to help you kind of focus. Okay. Okay. So that, and then I'll ask a question on something about how do you use mass spec to evaluate some sort of post-translational modification? How would you use it? There's only 3 post-translational modifications. We really talked about phosphorylation. We talked about that. Ubiquitination and glycosylation. So go to that slide because this is a proteomics course. Let's focus on the application of these things. So how many is that? 3? And then the 4th one will be on something related to, let's see, what do I want to, mutations, the interactome. What is, something to do with interactome? How do we evaluate? How do we find protein-protein interactions? Okay. And so that will narrow it down for you. What is an interactome? You got to know all that kind of stuff. What is an interactome? How do we use proteomics to identify interactomes? What is the significance of that? I'll send you out but then that'll narrow it down a little bit, right? Does that narrow it down?
Yeah.
Okay.
Thank you.
Yeah.
Thank you.
So, because there actually is a lot of information in all that, I think. If any of you know Garrett, let him know. Do you know, does anybody in here know Garrett?
Nope.
The guy that left?
Nope.
I've had him.
You never seen him before?
I've had him in plant bio before.
Yeah, he's in one of your groups. Whose group is he in?
Not mine.
Poor Garrett.
Garrett's in somebody's group. I got his name written down.
Sick burn.
What are you guys doing? Don't you know your
He's a bio.
Garrett's
major.
in Joe and Javier's?
I didn't even know that.
You didn't know it? What the heck? It's right here.
He's a bio major.
Hey, we're gonna talk about fish.
5 questions or 4 questions? There's like a last question. There's like a last chapter that we didn't talk about.
What, the last chapter? Which one? Control of regulation. Yeah, maybe and then a regulation one. Oh, she just left too. Do you know Haley?
Oh, no, I'm still here.
Oh, you're there. Yeah.
Yeah, it was Nate.
All right.
It was.
I didn't
Nate.
see you
Nate
out of
is
the corner
good.
of my eye. Who? Oh, Nate left.
Nate's fine.
Yeah,
He went to go find. He went to go find Garrett.
He went to go find Garrett.
Yeah.
So regulation, we talked about methylation. We talked about mechanisms. So we talked about the repressors, right? And how do we control things? Regulation. Something in regulation.
Okay.
So 5 questions. Let me pull up regulation real quick if you all have some time. And I'm sure you all do. Regulation. What do I want? Yeah, regulation. We looked at the lac operon and the idea of the operon system. It really informs you as to how things happen. So we talk about promoters. We talk about repressors. Okay, so understand the operon regulatory mechanism. We have repressors, right? And we have basically activators. So look at those slides. And then compare that to eukaryotes, which is different, because there we use transcription factors, right?
What's really the big difference between the two? That you can interact, that it comes back and interacts?
Yeah, so,
Makes loops.
well, in the one, it's not just the transcription factors, it's the methylation of histones and things like that. So, let's focus on, that's more histone. Let's just focus on the prokaryote version. Focus on the promoter, repressor, activator, and then also the third mechanism where you actually have things in the RNA itself that regulate translation. Tryptophan, the tryptophan one. Basically, right? It'll make a structure, and if you do have tryptophan, it'll bind to it and it will stop it from producing any more protein. If tryptophan isn't present, it will just continue to go on so that it can endogenously produce tryptophan. Does that work out for y'all? So that would be like slides 1 through 9.
Which chapter?
That's 26, regulation. And it could be anything on that, so understand all of that. Does that work?
Sure.
Sure.
Are you gonna be available today or tomorrow?
Come on, not at all. But what would you like? I've got to go. What time? Well, today, yeah, I have office hours today, 3 to 5.
What time? 3 to 5?
3 to 5, yeah. Okay, cool. You guys don't even know what the office hours are. That's
Alright, guys.
Yes, Tim.
Do you have any subscribers?
What's up?
Yeah, subscribers.
now only question is, will I remember what I told you 5 years?
I'm gonna troll
Did I
you.
write it?